Hi everyone, how you doing? And welcome to the actual lecture 2 for Biodiversity and Sustainability, WATS 1200. This lecture is all about graphs. It's a relatively short lecture, it's not very long at all, but I just want to give you an overview about some of the different types of graphs you're going to encounter when you're going through the rest of the course and also in the remainder of your lives, especially if you do something in any way sciencey. And also just to give you an idea about some of the things that a graph should do, some of the things they shouldn't do, and some of the ways to interpret them. Okay. In this class, I'm not going to ask you necessarily to draw your own graphs, given the logistics to do with the COVID restrictions and the fact that all quizzes are hopefully online and things like that. It makes it quite difficult for you actually to draw things. But you will be asked a lot to interpret graphs. So you'll be presented with a graph and then be asked questions about, okay, what is the relationship between this and that? Given that relationship, what can we say about blah, 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 blah. Okay. So although you need to get comfortable reading graphs and you need to get comfortable interpreting them. Okay. So without any further ado, let's get started. I will just do the share screen. Where is it? There it is. Okay, you should now be looking at this graph. So by the end of this class, students should be able to describe and distinguish 4 types of graphs. You should be able to interpret graphs to analyze patterns within real data. Okay, and the way I like to start out this class is by first of all asking, what should a graph really do? Well, the main thing a graph should do is convey the most amount of information as simply as possible and as quickly as possible. So the ideal graph is one that conveys an idea, a concept, gets it across very easy to interpret, and you can literally just glance at it and be like, oh, yes, okay, I see there is a relationship between this and that. This is very important because often some of the graphs you're seeing in scientific papers and even in sort of popular media, you should be able to quickly look at them and go, oh, yes, there we are, easy, I can interpret that. To that end, a few years ago, I had a competition with a senior professor at the University of Victoria in Canada, and we had a competition to try and find the worst possible graph we could on the internet, anywhere. A graph that was horrendous to look at, very difficult to interpret, didn't easily convey information, basically exact opposite of what a good graph should do. So we had a bit of a competition. We went away and just came back a week later, and we came back with the worst graphs we've come up with. This was my entry. Oh, give me a second. This horror just here. To even start to begin on what's wrong with this is difficult. So first up, let's just start with the fact that there's so much going on this page, it's impossible to actually ascertain what's really going on. Secondly, for some reason, they've made it not only 3-dimensional, when there's absolutely no reason for it to be 3-dimensional at all. It only adds confusion, it doesn't add clarity. Not only do they make it 3-dimensional, they also made it semi-transparent. And because it's semi-transparent and multicolored, you have all these weird things like, what is this green here? Is this green this? Or is it some other thing that's going on? Why is this magical brown here? What's going on with some of these pinks and weirdnesses all around the sides? So the 3D is bad, semi-transparent is bad, 3D and semi-transparent makes it absolutely awful. On top of that, it's just horrendous to look at. It's just ugly. The color scheme looks like some dodgy 70s kitchen. It's just bad. I thought I was onto an absolute winner with this one, thought it was an easy win. If you want to know where I got this graph, it's actually from an instruction manual for a piece of computer software. This was the graph in the instruction manual they gave as an indication of the power of this software and the amazing things you could achieve with this piece of software. Not surprisingly, this piece of software and the company that made it is now bankrupt and defunct, and you can't get it anymore. If this is the sort of thing they were showing to say, look how good we are, then I'm not surprised it's gone under. But I thought I was onto a winner. I thought I've won by miles. I am a genius. Turns out none of those were true because the person I had the competition with came up with this horror. So this, to this day, I still can't fully interpret it. we've got a whole bunch of things going on. First up, let's talk about the color scheme. Again, absolutely horrendous. It's just clashing and awful and just plain nasty. So we've got the bad color scheme. Once again, we've got the 3D situation. So this has got a 3D that goes from there to there. The issue with that is, when I'm looking at this point, what am I actually reading? Because this is 3D, should I be looking at where it crosses this axis here or here? Is it here or here? Or here or here? If it's here, then we've got all this extra space that we don't really need getting used, and we could shrink the whole thing down to this big. If it's here, which I think it is, based on the fact this says 72 and that's 72 and this says 70.8 and that's about 70.8. If this is where we should be looking, this bit is just distracting and in the way. It's rubbish. Also, the perspective on it is just all over the place. It's awful. Then we have to look at, okay, what are we actually looking at here? So when you try and interpret this, this is a graph of the age structure of college enrollment. This, I'm assuming, is the under 25s. This is 25s and over. This is what the year was. It's just awful. Even after going through this multiple times, every time I look at it, I have to argue with it and squint and stare, and I still can't exactly get it right. So it's bad, and as a result, Brad and Holt won. So remember, when you're making a graph, you want to be trying to convey the most amount of information as quickly as possible and as simply as possible. Those two examples are then two of the most terrible attempts at doing a graph that I've ever seen. From your perspective, when you're looking at interpreting a graph and when you're trying to understand what it's saying, it's often easiest if you break it down to these simple steps. The first thing we say is, well, how do we approach a graph? How to approach a graph? First question is, what type of graph is it? We're going to go through the major types of graph that you'll come across, the major types of ones that we'll be using in the later part of this lecture. But the first thing you should look at is, what type of graph is it? Secondly, you should ask yourself, what do the two axes represent? The axes basically represent the sides of the graph. What are they saying? Is there a label on them? Are they numbered, etc.? What are those axes? What am I looking at a relationship between? Okay. Number three, is there a pattern or trend in the data? Can you see how is this data relating to another data, any other piece of data? What's the relationship between one and the other? Given this, what can you conclude from the pattern? If you're looking at the relationship or between two factors, is it positive? Is it negative? What can you then say? And finally, what is the strength of the conclusion you're drawing? Okay, I'm going to go through that on the next page. I'll show you how we do it. So this here is a scatter plot. It's a line graph. On a scatter plot means it's got these little happy dots. The line graph, this is the line that's drawn through the points, and it represents what we call the line of best fit. This is showing there the general pattern. So if we go through our processes of how we approach a graph, the first thing we do is we look at it and we go, oh, yep, that is a scatter plot stroke line graph. We've got the two pieces of information on it. Next question, what are the axes? Well, on the x-axis, which is this one here, we have elevation in meters, so height off the floor. On the y-axis, we've got the mean number per pool. So we're looking at the relationship between how high a pool is and the mean number of individuals of a particular species. If we read the caption, it says, well, this is the relationship between total abundance of macrobrachium in the years 1988 to 2002 and elevation in Cuebra Prieta, which is sides of a series of mountains. What we can see is we've got our elevation on our x-axis, the mean number of individuals per pool on the y-axis, and we've got this line going down. So when we ask ourselves our next step, step three of, is there a pattern or a trend in the data? We can say, well, yes. It appears to be that as you increase in elevation, the number of individuals in a pool goes down. Okay. What conclusion can we draw from this pattern? Well, there's a negative relationship between height and individuals per pool. We then might extend that further and say things like, well, why would that be? If you're going higher up the slope of a mountain, the air is going to get thinner. It's going to be more isolated. It'll be colder. It's generally going to be a more exposed landscape potentially. As a result, that might be why we're getting a reduced number of individuals per pool as we increase in elevation. The next step is a little bit trickier. It's what is the strength of the conclusion? So the last step is what's the strength? The strength is based on two things. One, the steepness of this slope, if there is one. If it's really steep, like if it was going down like this, that would tell you, oh, this is a very strong negative relationship. This is definitely a negative relationship, not as strong as it could be, but it could be a lot weaker. The second stage is how tight are the points against the line? In this case, they seem pretty tight with the exception of this weirdo here. It generally appears that this is a pretty strong relationship. It's clear, the pattern, and all the points are reasonably tight on the line. Often in ecology, you'll see what are affectionately referred to as shotgun blast graphs. And this is where you have your x-axis and your y-axis, and then you have a whole bunch of points. In fact, I'm going to do this properly. Where's our screen? Pointer options, pen. So we have our x-axis, our y-axis, we have all, and then, but on the graph, we have all these points absolutely everywhere in a shotgun blast pattern. And then somebody has very optimistically just drawn a line through it like, yeah, that looks good. You'll see a lot of those in ecology. And so I'm asking about the strength of the relationship. We're not just wondering about this line here. We're also interested in how tight these points are to the line. In our case, on this figure here, this relationship with all the points nice and tight is much stronger. We have more confidence in it than this relationship here. Okay, let's go back to our laser pointer and hope those all disappear. Okay. The next type of graph you look at is a bar chart. And on a bar chart, the crucial bit is the x-axis. So this piece down here is categorical, as in it's just different types. So on this graph here, let's look at the caption first. This is actually from one of my papers. I'm looking at the log 10 abundances of 2 marine zooplankton with or without predators under ambient or elevated CO2 levels. The black bars, the null model, show what we'd expect if the abundances of the impact of predators and PO2 acted independently. This is from my paper in 2018, Global Change Biology. So let's go through our various steps. What type of graph is it? Well, first of all, we've got a categorical x-axis. A predator, no predator or predator is categorical. It's either predators there or it isn't. So we've definitely got a bar chart because we've got categorical x-axis. Next step, what are the axes? Well, the x-axis is predators or not, and the white bar and the grey bar is this is ambient CO2 and this is elevated CO2. And on the y-axis, we have the log abundance of a calanoid copepod or a log abundance of a Plagiomata. Plagiomata is a baby crab, basically. So we know what our axes are. Is there a pattern in our data? Well, let's have a look. First of all, we'll look for these copepods. What we see when we have a look is we see, okay, let's have a look at the effect of CO2. So we'll compare CO2 in the predators. So this is when we haven't got a predator and we've got ambient CO2, we get a nice big bar. When we have no predator but elevated CO2, so increased CO2 in the water, we have a reduction in the abundance of these calanoid copepods. The abundance drops from here to here. Now let's have a look about the predator effect. So when we've got a predator present in ambient CO2, we still have a lot of these copepods. We actually see a bit of a drop based on when there's no predator. But when we have CO2, elevated CO2 and a predator, we have a very large drop. So what we can say from this, for this species is, well, adding a predator in ambient CO2 reduces the number of individuals, reduces their abundance. This is probably because they're getting eaten. If you add just CO2, if you increase the level of CO2, you also get a drop. You get a reduced number of these calanoid copepods. But if you add a predator and elevated CO2 at the same time, you get a really big drop. So it drops from here to here. If these two factors acted independently, we would expect this abundance here to actually be this black bar. But because it's much lower, it shows that the effect of increased CO2 and predators is combining together, and it's what we call an interaction, and the effect is much greater than we would predict. Okay, so we can start to interpret these things. If we have a look at this crab, these little crabs, we see, first of all, we have just very few crabs generally. Adding elevated CO2 doesn't seem to have any effect. There's no difference here. Adding a predator doesn't really seem to have any effect, same as it is here. When we add a predator and CO2, we get a big drop in the abundance of these little crabs. So again, what we can see is the effect of a predator and CO2 is interacting to reduce the abundance lower than what we'd expect based on the null model. So we can start to interpret these graphs. Next question. What can you conclude from the pattern? Well, our error bars, which show our the estimate error around our actual estimates, are pretty narrow. So this means we can be quite confident in our results. And we can see this is quite a big drop. So it looks like there is a big effect. So generally, what we can say is predators seem to eat copepods. Increased CO2 is bad for copepods. Increased CO2 and a predator is very bad for copepods. And we can see in crabs, CO2 on its own doesn't have an effect. Predator on its own doesn't have an effect. Both together, big effect. So we can understand what's going on. Okay. Next type of graph we'll look at is a histogram. Now, a histogram at first looks very similar to a bar chart. But the big difference between a histogram and a bar chart is this. The x-axis is a continuous variable. And also the y-axis must be frequency or number of something. So when you're looking at a histogram, this must be continuous. In this case, it's length. So length goes from, in this case, 200 to 2,600. Remember, on the bar chart, this was just categories. So it was one, this, that, the other. Also on a histogram, the y-axis must be frequency or number of times something is counted. This is a figure taken from a paper produced by a student of mine, looking at the impact of this little fish. It's called Gambusia holbrooki. It's a highly invasive small poecillid, especially in Australia. And he was looking at the length of macroinvertebrates, so bugs in the water, when you've got these present and when you haven't. And what you see is that basically when Gambusia are present, you just get a lot fewer. All of these bars are much smaller, and there's a slight shift in the distribution of the species. The crucial bit about histogram versus a bar chart, histogram must have frequency, must be continuous, not categorical. Okay. So when we look at the difference here, so we've got things like, this is taken from, these two graphs taken in the same paper. So difference in a bar chart and a histogram, categories, continuous. Also, this is abundance, this is frequency, okay? The last sort of graph we're going to look at is a box plot. This is one of them here. These are wasted box plots, as in they have a waste, not they've been wasted. And box plots are a little bit more difficult to interpret, but they contain an awful lot more information. So when you're looking at a box plot, these two lines here represent the range of most of the data. You do have outliers out here, but this is the range of the data. This bit is what we call the interquartile range, which means 50% of all the data points lie within these lines. These wastes represent the middle 25% of the data, and this is the average here. So when you look at a box plot, you not only get an indication of what the average result is, you also get a lot of information about the spread of the data. Okay, and so let's go through our process as we did before and look at how do we interpret these graphs. So first of all, what type of graph is it? Well, it's a box plot, we just said that. What are the axes? Well, on the x-axis, we have fertility treatment, and we've got low, mid, or high. On the y-axis, we've got relative yield total. So we're looking at what happens when you fertilize something, how much of something gets produced. What is the yield? And what we do is when we look at the caption, we see this is a graph of the amount of plant biomass relative to a monoculture. So a monoculture is when you only grow one species. Relative yield produced by diverse plant assemblages under soils of low, mid, and high fertility. The line at 1 indicates the yield of the monoculture. Okay, so what we're doing is that as we increase fertility, as we increase the amount of nutrients, we fertilize the soil, we see generally we get a higher yield of plants. But what's interesting is when we have low amounts of fertility, the average plant yield is actually less than this line, which is if we did have a monoculture. At high levels of fertility, this average is actually higher than if we had a monoculture. So we've got an increased fertility. So you can get an awful lot more information out of these. Okay. So as I said, by the end of this class, the students should be able to describe and distinguish 4 types of graphs. They are line graph, stroke scatter plot, bar chart, histogram, box plot. You should also be able to interpret graphs to analyze patterns within real data.